Easy Cancer outputs. If Output Files option is selected for each seed, Easy Cancer generates 4 outputs. In the seed ancestor results output, you will find the total population per generation, which can be used to show the population dynamics and to determine the time required to reach a defined population size. In this output, you will also find the number of descendant cells for each ancestral cell of the initial cell population in each generation. A run starting with 1,000 cells will have 1,000 columns, each column corresponding to one ancestral. Runs with different seeds can represent different evolutionary stories with growth dynamics that are distinct, with some seeds leading to an early growth of the population and others not producing any growth at all. In the seeds gene mutation results output, you will find the number of cells affected on each gene in 1 or 2 alleles in each generation. Similarly, in the seeds events mutation results output, you will find the number of cells affected in each event in each generation. Easy Cancer also creates an output called parameters, in which the initial parameters of a specific run are saved. If Output Files option is selected for entire simulations, Easy Cancer also generates 4 outputs, containing summarized data from the final generation of each seed. In the automatics ancestor results output, the number of descendants for each ancestral cells in the final population of each seed is saved. Additionally, in this output, the total number of divisions of a specific run is saved. The generation in which a stop condition is reached for each seed can be used to construct a Kaplan-Meier-like plot in which a large number of runs are presented in a form that can be easily recognized by cancer biology. Here we show runs with different events per division rates. An increase in the event load from 5 to 10 events per division increases the proportion of runs that develop easy tumors. Further increase in the event rate reduces the number of generations required for 50% of the runs to produce an easy tumor. In the automatics gene mutation results output, you will find the number of cells affected on each gene in 1 or 2 alleles, in the final population and for each seed. In the automatics events mutation results output, you will find the number of cells affected on each event in the final population and for each seed. Data from this output can be used to analyze the frequency of alterations in a given event. Different events affecting a given gene can also be pooled. For example, in a setting with 6 genes and 7 events, increasing dominance, as expected, increases the frequency in which this event appears in the final population. Final frequency is positively correlated with cell fitness, compare gene 4 and 5, and with probability of events, compare gene 5 and 6. A gene can have more than one event. For example, gene 3 has 2 events, and event 2 can allelically complete event 1 and vice versa. Therefore, the frequency of this event is higher when compared to gene 2, event 1. An event can also have more than one gene. To model copy number variations, event AB impacts gene/event A and B, but not C, and therefore increases the frequency of these 2 genes without affecting event C. If event B can also occur independently, then the frequency of this event is the sum of the 2 events to model, for example, deletion in one allele and mutation in the other. Needless to say that individual runs produce quite different results due to the stochastic nature of easy cancer, but the average frequency behaves as predicted by evolutionary biology. Finally, in the sequence each cell output, the single-cell genes in the 2 alleles are represented by 2 independent lists in a binary form, in which 0 represents absence and 1 represents the presence of an alteration in the gene. From this output, it is possible to infer phylogenetic trees using specific software, as shown in these examples, resulted from easy cancer simulations. These are only some examples on how to use easy cancer to explore key questions of the evolutionary aspects of cancer formation. A guide and spreadsheets to help you construct these figures are available at Labs in our website. Now you can start your own simulation. Feel free to contact us for any doubt or suggestion.